All right. I am pleased to say that we have Sherry Flint Garcia presenting on some of her work on Maize heirlooms. Uh, a little, dramatic, kind of getting, a USDA talk going again with current events. But happy to have you here, Sherry. So with that, I'll go ahead and stop talking and let you get started.
All right, great. So I'm going to give a little bit of an overview of what my group does. It's going to be a bunch of short vignettes. All my students gave committee meeting updates or seminars on campus in the last month. And so I put them all together. That's why you'll see different styles of presentation and slides and things. So we're gonna roll with it. Okay, so the way to a man's heart is through his stomach. It's my opinion that corn in the US has a PR problem. When people who are in the agricultural sector think about corn, we all know and love corn and respect corn and see the value of corn. Farmers see the value of corn. But if you talk to the average person in a metropolitan area, maybe beyond the average person, people think negative thoughts about corn. High fructose corn syrup. It's a subsidized crop. It's bad for the environment. It takes all this nitrogen and contributes to environmental effects. It's just bad. It's all GMO. Everything's bad. And so the way that people care about things is through what they eat. And so this is where my research is kind of focused. I do not need to explain to this group about the history of corn. I only want to show that, I have experience working with teosinte inbred lines, and now my real focus is on land races, which is the scientific term, but we prefer to use the word heirlooms. You'll see why in a little bit. And the whole reason for this is all about the consequences of domestication and breeding, all this artificial selection. We've lost allelic diversity, especially for genes that were important in domestication and improvement, about 1,000 genes out there. And it's all really been driven by yield, especially more recently by commercial breeding programs, that a lot of allelic diversity has been lost in the name of yield. And just like a rose without thorns, sometimes you lose the beneficial traits like the aromas and things like that. And that's my argument about corn, is that we've lost a lot of really neat things along the way. And if we're going to get people to love corn to the extent, even remotely to the extent of what we do, we need to get them excited about corn again. So you can see these patterns about how there's been selection for in breeding programs by looking at the inbreds from around the world. This was the Ames panel from Ed's group and our group back a long time ago now already, where we genotyped 2,800 inbred lines from the germplasm collection. And just by relationships among the markers, you can see the clustering that's gone on in the sweet corn breeding, the popcorn breeding, the two sides of the heterotic pattern that make our number 2 yellow dent of the tropical germplasm. But my argument is that there's a lot of other corn that people eat that's not represented by breeding programs, and we should change that because there's a lot more to corn. So I'm really interested in grain composition diversity, not just in the basic compositional traits like protein, starch, and oil that are easy to measure and cheap to measure, but unfortunately, the things that really matter in flavor, textures, and aromas are the really expensive metabolites. And so that's where I'm trying to dip my foot into without any substantial funding at this point, to look at the metabolomics of flavors and textures and aromas and things like that. So I have a lot of projects that are focused using the diversity and the heirlooms or the land races that I'll touch on in the next, hopefully less than a half hour. I have a lot of slides, so we're going to have to cruise through these really quick. So the first project is one that's spearheaded by my graduate student, Melissa Draves. This is part of a NEFA-funded project with Jim Holland at NC State, Joe Gage at NC State, Jim's with USDA, of course, and with Jacob Washburn here with USDA at MU. And so we're characterizing the heirlooms of the United States. And again, heirlooms are land races. Scientifically, the term is land races. The romantic term that we need to get people excited about because is heirlooms, because they're passed down from generation to generation. They have value, they have charm, they have character, they have the riz. And so, heirlooms are these populations of heterozygous, heterogeneous plants where if you had 1,000 plants, they're all presumably going to have some variation from one plant to the next. They're not the same. They've been open pollinated and adapted to a specific region. They may have been moved from their region, but it's got some form of adaptation to some specific environment. In the past, they've been used for food and livestock feed, but modern day uses, there's a lot of interest in craft applications, culinary, beverages, natural food dyes, and things. And so we'll touch on all these as we move through my talk. So the objectives of Melissa's project and the Heirlooms, U.S. Heirlooms project is to create a comprehensive, systematic genotypic and phenotypic data set to characterize all these heirlooms from the U.S. Because there is no such thing. There is the heirloom or the land races of Peru, the land races of Chile, the land races of Japan even, but there is no such collection of information systematically characterizing the U.S. heirlooms. And then once we have all that information, we can see all the different relationships that are in the germplasm and identify targets for culinary, which is my main interest, breeding efforts in our group. But make it useful for everybody else. Because if you go onto the GRIN database and you type in land races and part of their search, you'll get a list, and sometimes there's no information on them. There's very little phenotypic data. If you want to use it for some particular purpose, you have almost no starting place of how is it adapted? What's the grain composition like? How are you going to use it? So the experimental design, we had 2 locations in North Carolina, Missouri, the last summer and this summer. We characterized nearly 1,000 heirlooms, and that also the experiment contained repeated checks and 2 entry checks. That had a partial replicated design of 1.25x. And you can see where the distribution of all these heirlooms are coming from. The vast majority are from Arizona and the desert Southwest. Colorado is kind of artificially high because it's where the germplasm collection in the mountain is for the long-term storage. Missouri has a lot of them. North Carolina has a lot. South Carolina has a lot. But there are some states that don't have any heirlooms that are in the collection. But we've evaluated these now for 2 whole years in each environment then, which is a location year. We have about 1,280 rows, which represents about 1.25 acres. And it was a lot of corn, folks, and we are done with the field trials. Thank goodness. Finally, we have all the field data done. We collected all the basic things as you'd expect: flowering time, plant height, ear height, leaf length and width, tassel architecture, some traits that were more relevant to growing heirlooms, like if the ears drop and hang, that keeps the water out. Husk coverage keeps the insects out. Some different characteristics there. And this project was very large. We pollinated 3 plants per row. So this was a major pollination effort and a lot of data collection in both of the locations. And so again, thank goodness we are done. Just by looking at the phenotypic data and doing cluster analysis, PCAs, both, for example, the Missouri 2024 and the North Carolina 2024 data, you can see that the heirlooms are self-arranging themselves based on the phenotypes from about north to south. The reddish colors are farther north, and then the more yellow, blue, and then green and purple are farther south. And so in both locations, we're seeing what we're expecting, that the principal component 1, which you can see in almost any kind of a diversity analysis based on markers or phenotypes, is really driven by that adaptation to day length. Principal component 2 seems to separate the desert Southwest things from other things a little bit more. We're going to, of course, genotype and see how the genetic data correspond then to the phenotypic data. Melissa then took the data from the the Missouri 2024, and then she color coded them based on kernel types. And again, you can see that the sweets and the flints are much more, the principal component 1, more northern adapted, and then the dents are much more southern adapted. And this is also in line with historical data. And this is another reality, another check to make sure that we're actually seeing what we expect. We are in the process now of finalizing our automated imaging pipeline for phenotyping ears and kernels. We are very well aware that the Wisconsin has a imaging pipeline, but it doesn't do some of the things that we want it to do. So we're trying to now incorporate color, automated color into the system. And so we had a hackathon back in January, back in the good old days. And we got through almost all of the processing of the figuring out the kinds of pieces that need to get put together so that we can take our data, mask out the background, align the ears, so with the central axis, et cetera, et cetera. Ultimately, we're taking for each plot 3 scans, intact ears. After the kernels are shelled off, we have the cobs and the kernels. And you can see the traits that we are realistically going to be able to collect using this system. And so these are all data that are going to feed into the GRIN database so that when people want to go into GRIN and find some heirlooms for whatever purpose, they will be able to find out what kind of grain and everything, what are the measurements, what are the adaptations and things like that. So as a quick check, so then we're also looking at grain composition. In this case, we're using near-infrared spectroscopy to estimate protein, starch, and oil. And you can see the distributions. At this point, we only have about, let's see, this is probably about an eighth. You know what? It doesn't matter. We're just working through, at this point, we've gotten through the Missouri 2024 yellow kernel set of everything. We wanted to have different calibrations for yellows, whites, and other colors because they are well known to have the calibrations break down when you go outside a certain calibration. So you can see that we have a nice distribution for each of the traits. We check to see that the expected relationship, the negative relationship between starch and protein exists, and that's very perfectly aligned. Everything's great. But we found a couple interesting things. What's going on with these extremely low starch and extremely high oil samples here? It's actually just one. It turns out to be a sweet corn, and that's not surprising. When you don't make starch, that makes starch very low. And then that inversely makes oil really high. And I expect these that are on the lower tail of starch and the higher tail of oil to also be sweet corns. This particular accession, when you look in GRIN, you can see that this is a sugary type of sweet corn heirloom. The germplasm collection has lots of samples of when they've grown it over the years. And for this particular one, I don't remember the year that this picture was taken, but this is an earlier representation of that heirloom. But then this actual sample that we got is clearly contaminated with shrunken 2, not just the sugary. So shrunken 2 has a freakishly different morphology compared to the sugaries. And I think we're seeing that is representing itself, showing itself here in the starch and the oil. Because shrunken 2s are a modern invention, if you will, a discovery of a mutation in the 1970s, we don't expect all these heirlooms in the collection, the sweet corns, to have shrunken 2s. This is something that we found kind of interesting. So of course, we're only in the second year of our 3-year grant. And so we have, of course, collected samples for whole genome sequencing, all these different plants, these accessions. 10 individuals were going to be genotyped individually using whole genome sequencing approach. We also collected for a couple of years in a row RNA at the seedling stage for all of these. And so we are well underway. North Carolina is working on the genotypic data, processing all that. And ultimately, we're going to be looking at relationships and doing GWAS to identify targets for our culinary purposes. Speaking of culinary purposes, we have a collaboration with Yole Tortilleria in Kansas City, where we're exploring heirlooms for unique corn flavor profiles. They've been bringing corn in from Mexico, and it's really expensive for them to bring in the corn to make these custom tortillas. And so we're trying to find if we can make heirloom corn in the US and grow local and all that stuff. I think I'll just move ahead on that. We also have a tortilla, stone mill in our lab, so we can do our own tortilla work. But we have an active breeding program working on tortillas and colors and many other things. And this is just an array of 16 tortillas, batches of tortillas that we made this spring. And some of them have really pretty colors, and some of them are ugly, and some of them tasted great, and some of them tasted terrible, as one would expect. And so this work is really spearheaded by Nancy, a research scientist in my group. She has the touch on a lot of things when it comes to the artistic aspects of corn. So we have a lot of different breeding populations that we have crossed different combinations to make, for example, flinty populations or flowery populations or popcorn populations, where oftentimes we'll take something that's adapted to South America, for example, from Chile, and cross it with something from Europe or something from the Southwest, from South America, making new combinations, bringing together genetics to try to improve the vigor and the quality of some of these heirlooms, and adapting them then to the US, including color. And so it turns out people like color and patterns. And so we have blues and we have reds and we have spotties and we have stripies and we have all different combinations of things. So these are all populations that we've had in the nursery now, I think, for 3 years in a row. Boy, that's a lot of corn. Speaking of all the colors, all these different colors come from different layers of the kernels. So the outside layer that always gets stuck in your teeth when you eat popcorn and sweet corn, that is the pericarp, made of different kinds of layers. There's almost exclusively a single layer of aleuron tissue that accumulates different types of pigments than in the pericarp. And then, of course, you can get pigmentation in the endosperm itself. And it's the combination of these pigments and these different layers that can give you different colors and things like that. So the phlobophene, the anthocyanins, and the carotenoids. And just like in elementary school, you can mix and match your paints. So in order to make pink, you just have a very small amount of red. And so you can make a very light layer of pigmentation in the aleuron, and with the most of its white underneath, you get a very pale pink. But by increasing your pigmentation in that single layer, you can get things that are much darker. And you can mix your paints together. You may have been around for Jim Holland's talk back in the spring when he talked about how Oaxacan green dent is not from Oaxaca. And you may have seen this particular combination of images already. In order to get green, you just have a thin layer of bluish color with yellow underneath and maybe some other pigments mixed in there. So Oaxacan green is neither from Oaxaca, nor is it really green. It's just a combination of blue and yellow. And then we have some very unique combinations of colors that are what we see as brown, but it's actually different layers of kind of an orangish color and a magenta underneath to give you that brown, which is fairly unique. You can see when you slice kernels, these are all things that Nancy has taken upon herself to learn. I am not the microscopist you're looking for. So Nancy is the person who's figured out how to do all this and has been taking all these amazing images. But you can see even in striped germplasm, the combinations of the pigmentation in certain layers of the pericarp. We even have things that have different layers of spots and stripes, and you can see how the spots and stripes come from different parts of the kernel as well. The spot being here with coloration, not on the right. You can see the different colors of the stripes. So why do we even care about pigmented corn in the first place? All these anthocyanins, flavonols, phenolic acids, and things like that, they have potential health benefits for human beings. And we're also trying to replace synthetic food dyes. For example, Red 3 and Red 40. Red 3 has been recently banned in California. We're trying to also find a replacement for caramel brown coloring because it has suspected carcinogens. And if you know anything about me, you know that I love my Diet Coke. So the idea of when I go to committee meetings with the food scientists on campus, carrying my Diet Cokes, one in each hand, and then probably a little tiny bag of candy as well, I'm horrified to be the example in the room of how, see, look at Sherry and how terrible it is. In addition, the pigmented corn offers some value added, especially in the pericarp, where if you roll up to an ethanol plant, the first thing they do is they peel off the pericarp layer and it's just trash. So if we can put a lot of pigmentation in the pericarp, then we can add value to the crop. Okay. So we have been on the hunt for heirlooms that have extreme pigmentation, and I'll just focus on the black, deep purple, dark red, whatever you want to call these colors, looking for combinations of either thick pericarp or intense pigmentation. Our food science collaborators on campus, they have the milling equipment to tell us what kinds of fractions, the coarse grits, medium grits, fine grits, and the different pericarp yields, and they quantify things for us. And so we get what the real world milling would look like if we were going to make products out of this. And so there's a lot of variation in kernel types and things, and we've been exploring these. By far the best so far has been Maize Morado, and this is something from South America. Unfortunately, so it has the highest anthocyanin pigment concentrations of anything that we've screened so far. Unfortunately, if you plant 20 kernels of Maize Morado in Missouri, you might get 15 back. It's terrible. You can't reach the ears. It's just bad news all the way around. So in addition to screening all the different accessions that I've been talking about in the previous slides, we're also introgressing these color genes from all of our different donors, not just Maize Morado. Maize Morado is the farthest along in the collection, but other things, not just purples and reds, but also the Aleuron colors, blues, and other things like that, and pinks. We're introgressing those into different XPVP backgrounds with either white or yellow endosperm or stiff stock or non-stiff stock heterotic patterns with different color combinations from about 25 different donors. And so this is a fairly large program. This is the one thing that is my section of the field. And so when I want to get away from people, I just go and I work in this. So the current status, the most advanced things we have are BC5s. That would be the Maize Morados because we started those back in 2021. We have advanced a small subset of these as a proof of concept that we've solved a couple times, and we're going to try to make some hybrids this coming summer and test them the following summer and see what the yield potential and stuff like that might be. Okay, so let's see, how do I transition here? This is all the pigmented corn. Earlier on, I talked about how some of these pigments and things are not healthy if they come from petroleum products. But so we're trying to find natural sources. And one of the reasons is that when you're cooking corn, you can create acrylamide due to the Maillard reaction. So this project is Sarah Fitzsimmons. Sorry, she got married a couple of years ago, and not all the slides have made the name change yet. But Sarah's project is looking at the genetic basis of asparagine as it relates to acrylamide formation. So the Maillard reaction is when you cook, think frying potatoes or corn chips or something like that, you get all the yummy, yummy flavors and aromas and colors and things like that, which is why we cook our food. But unfortunately, we can also get asparagine reacting with some of those byproducts, making acrylamide, which is a class 2 carcinogen. So let's boil that complicated equation down into the idea here that you have reducing sugars that can connect together to make starches. And the amino groups, they make all the yummy, yummy byproducts of the Maillard reaction. But if you have free asparagine floating around in your food, there's another set of chemical reactions under the right pH and get different conditions that can make acrylamide. And so this is the part that's no bueno. So in other crops, they've dealt with this in different ways. Potatoes and wheat are major staples. I already mentioned potato chips and French fries and things like that. In the case of potatoes, they can alter both the reducing sugars, but also the asparagine as well, using asparagine synthetase. And then likewise in wheat, they've been dealing with this. So what about corn? It's only a matter of time till someone starts to pick on poor corn again. So it turns out that in corn, there are 4 asparagine synthetases, as in 1 through 4. And so we were excited about some of these things, and we have a lot of different approaches in our group to try to study this. So one of them naturally is a quantitative genetics approach, just GWAS. So we looked at the Buckler-Goodman panel of 282 lines, and you can see that there's extreme variation, 20-fold variation in the amount of free asparagine in 2 years and 2 locations of growing this out. Sarah is co-advised by Ruthie Ungalovici here at MU, and so Ruthie specializes in amino acids. So just this distribution itself tells us that it's biologically possible to make low asparagine corn. Many of these really low lines here are really old and they're not elite. And so we want to try to make modern corn a little bit better. And this also suggests that there's probably genetic control since the heritability is fairly reasonably high in this. I don't remember the value off the top of my head. So we did GWAS. You can do GWAS for amino acids in different ways. You can look at the absolute levels of asparagine, for example. You can take asparagine relative to the total amount of amino acids, what proportion of the protein is made of asparagine. Or you can look at relative to different families. And so what's the asparagine compared to the aspartic acid family as well. She used different models, looking at different levels of population structure control and different things. And in summary, we've got a lot of SNPs, about 154 SNPs. And when you look at the confidence intervals, which are plus or minus 100 kb on each side of the significant SNP, we have about 4,200 and something genes. One of them was kind of exciting. Amino acid permease 6 was our biggest one. The only thing I want to tell you right now is asparagine synthetase is not one of these candidate genes. None of the 4 came up there. So we were kind of like, that's interesting. So we took another tried and true method. We did QTL mapping by crossing the lowest asparagine line with the highest asparagine line and just made a QTL population. And 2 reps growing out in Missouri a couple of years, or last year, I guess, got the grain, did the amino acid phenotyping. We did an agriplex SNP chip with about 2,000 something SNPs on it, and QTL mapping. The 10 chromosomes are shown here. I want to point out on chromosome 1, we have this very large, a LOD score of 18, almost 19, which is ridiculous, controlling 30% of the variation in free asparagine. And it's right in the region of asparagine synthetase 3. So we are still working out, this was our very first pass, it's a fairly new result here, trying to work out what the confidence intervals there are, but certainly the asparagine synthetase 3 lies within that confidence interval for that QTL. So how come if we use the association panel and we can find a QTL, the big asparagine synthetase QTL peak, how come we couldn't find that using GWAS? So it turns out that asparagine synthetase 3, there are no SNPs in the data set. That's a head scratcher. 55 million SNPs divided by 50,000. There should be lots of SNPs within asparagine synthetase 3, but there aren't any. And for that matter, there aren't SNPs in any of the 4 asparagine synthetase genes. These are SNP deserts. Accidentally wrote deserts or desserts last night, and I thought, nope, I'm going to change that back to a SNP desert. So Sarah's exploring different steps here. There's other ways to look at the SNPs that maybe didn't make it into the HapMap3 data set, trying to see if they got filtered out for some reason. The 2 parents were building genome assemblies. And so we can see what's the difference. Maybe there's some presence absence, some other reason, structural reason for whatever reason that asparagine synthetase 3 is not showing up in the GWAS, but it is showing up in our QTL. The next steps there is we are validating ASN3 by getting a knockout, and some CRISPR constructs as well were designed by Wisconsin Crop Innovation Center as part of the CIRCA project. I will not have any time to talk about CIRCA today, so this is coming soon to a research organization near you. So the whole goal of Sarah's project is to be able to develop low acrylamide corn products so we can have our chips and eat them too. So the next project, speaking of eating your corn, now let's talk about drinking your corn. Zach's project is focused on flavor components in whiskey. He's co-advised by David Braun, so he has kind of also some background in carbon sugar accumulations in the leaves and carbon partitioning and things like that in the plants. That's the reason for that there. It should actually be a big bottle of whiskey. So the history of corn and whiskey, back in the day, they used to use local varieties. This was back prior to Prohibition and prior to hybrids in the US. Farmers used to grow what they had, and so everything was very local. But then about at the same time, we had Prohibition in the US, and we also had hybrids being developed in the US. And so those 2 things went hand in hand, and all of our beautiful heritage corn whiskeys have been gone. So we're bringing it back. Gary Heingartner is a craft distiller halfway between Columbia and St. Louis. And he came to us a couple of years before the pandemic and said, we've got this blue corn and there's a red corn. And they're very different. Here, taste the whiskey. I'm not a whiskey drinker, but I tasted them and they are very different. You know, the people who talk about wines and the bouquets and all those things, they do the same thing with whiskey. And so we've been working with Gary now for a number of years trying to look at all different kinds of corn and see what might be good for him. Zach's project, which I'll talk about here in a minute, trying to develop a lab scale process to explore the metabolites and things like that in the whiskeys. So grapes and wine serve as a model for this, where the precursors are in the grapes. You know, you grow certain grapes to get the different flavors. They've come up with different flavor and volatile compounds. They are worked out quantification and everything, and everybody finds wine to be socially acceptable and things like that to do this kind of analysis. And so we're going to make it into corn whiskey. And so there's been very little research on the metabolomics, but this Arnold paper from Seth Murray's group back in 2019, they looked at terroir and varieties and metabolomics underlying that. Luckily, there's a lexicon already for craft whiskeys or whiskeys in general. I don't think that I'd ever want to drink a whiskey that tastes like barnyard. I grew up on a dairy farm, so I don't think so. But it's interesting. I could talk about this for hours. So Zach, his first project was to develop a method where we can start off with a single ear of corn, do the whole grind, cook, ferment, and distill process to separate out the different components, the good, the bad, and the ugly. The good is the part that you want to drink. The bad makes things taste bad, and the ugly is what can poison you. To try to figure out which parts to take off the fractions, and analyze those for metabolomics. And so the amount that we get off from a single ear is roughly a shot glass. So Zach has been working on that method and we're working on a manuscript now. The goal is to do untargeted metabolomics to find out the flavor metabolites. And so there's a combination of different technologies. I'm not going to get into this. I am not the metabolomicist you're looking for. Using a combination of SPME, which you can test the headspace and the volatiles in your sample, and mass spec and GC and all this great LC, all these things. I am not the metabolomicist you're looking for. But after deconvoluting all the different things, you can get a list of metabolites that are in your sample. And we're going to start off by taking the two parents of a population that Zach has made. This is Gary's red and Gary's blue that we know have different flavor profiles. And we can presumably find those combinations in the offspring of our population. Do untargeted metabolomics in both of the parents. We have several reps in several different years looking at the unfermented. So this is before the yeast actually do their action, but after the alpha amylase has broken down the starches. And then the final distillate in there, we're also going to do NIR on the raw grain so that hopefully we can find signals in the NIR and the raw grain, and we don't have to do all that distillation, that pesky alcohol business. Just kidding. So we're going to do the untargeted metabolomics in the parents. And then the population has just a standard biparental, cross them together, make F2 derived F3 families, about 200 families. They've been genotyped with the agriplex SNP array as well. We've produced grain from those the last 2 summers. And we're just going to do the targeted metabolomics on the mash or the raw grain. We're still working out the detail there. I'm going to advocate for the mash, just to tie it in with the untargeted in the previous, but we're going to do QTL mapping and find the metabolites that come out from these 2 different parents. Okay, so that's all the heirlooms. So, you know, we talked about, what do we even have in the germplasm collection? What colors do we have? Because we can make tortillas and we can try to prevent making bad things in our Diet Coke and our tortillas, and we can drink our whiskey and get excellent flavors. Something that's slightly different here is going back a little further in time. So Tom Burgess is a new graduate student in the group. He's co-advised mostly in Jim Birchler's lab, and you'll see why here shortly. But he's working on a de novo domestication project using teosinte. And so we're going to talk about, he's just finished his 2nd year. He's in his 3rd year now, I think. The math is hard for me today. So Tom is working on trying to figure out his project plan. He just passed his comp, so yes, he's in his third year. But we're trying to find a suitable teosinte to work with, and it's quite the saga. If you've worked with teosinte in the past, there's a reason why people don't work on teosinte. It's hard, people. So we have access to something called, oops, I should have said that before, Northern teosinte. Okay, so Tom's broader research goal is de novo domestication. This is my slide kind of summarizing his comprehensive exam proposal. Basically, how close can we get going from teosinte to a primitive maize by modifying those handful of domestication loci using our modern technologies instead of waiting around for 9,000 years, because Tom does want to graduate in his lifetime. And so, of course, the main traits that we're concerned about are apical dominance. Oops, this is supposed to be naked kernels. I didn't copy very well. And then inflorescence architecture. And we have some candidate genes and things like that. And that's not the topic of today. Maybe Tom will talk about this someday soon. But one of the first hurdles was figuring out what teosinte we were going to work with. So we tried with just regular teosintes, the fruit case, the seeds are tiny. You got to get the embryos out to make the callus. It's really hard. In addition, you have the photoperiod sensitivity and the fact that you can't pollinate teosinte manually. It's got a whole host of problems. And so at the Maize meetings this last year, Tom met Bill Tracy, and they were talking about Northern teosinte, which was developed by Walt Gallinant. Walt Gallinant was in Massachusetts, and he basically crossed a teosinte from Guerrero, so we don't really know if it was Mexicana or Parviglimus, and he crossed it with Gaspé flint, and then back crossed it 5 times to teosinte while selecting for flowering in Massachusetts. And so these are just some pictures showing Parviglimus in the field in Missouri. This is probably about, I can see the Gaspé is right there with a tassel. So this is probably about a month after planting. This is Gaspé in the field in Missouri. And then you can see the Northern teosinte on the right side of the screen. And it's got this really weird and interesting characteristic where the main stalk will develop and look like Gaspé flint, this little runty little thing. It won't make pollen, but then it makes all the tillers, which will grow up and become tall like the teosinte. And so the major advantage of this material is that it will actually flower in Missouri, almost, you'll see the data here shortly. Amazing. So Tom went through a process of phenotyping Northern teosinte in a bunch of different environments for a bunch of different traits, all the different teosinte relative traits, naked kernels, the flowering time, number of tillers, seed shattering, and things like that, just to try to do some basic characterization. This has never been formally released as a resource, and so this is the main objective of this first project here. And you can see just by looking at the pictures, it is very teosinte. I have things that I call teosinte that's adapted, but I can tell that they're not teosinte because the kernels don't shatter. This is teosinte. It just flowers in Missouri. So he phenotyped it in the field here in a randomized complete block design, including Gaspé flint, Parviglimus, and Mexicana. He's doing just the Northern teosinte in the greenhouse and the growth chamber for various reasons under different growing conditions. And so for all practical purposes, I'm going to boil it down. It looks like teosinte. So in the field, which is all I'm going to talk about today, you can see it's just gorgeous. It's just amazing. You can see a comparison of Gaspé flint, which flowers very early. Northern teosinte flowers fairly early, flowering on the main stalk, but not actually producing pollen or silks that we've seen. Compared to Mexicana, Parviglimus didn't even flower in Missouri, so there's no data for Parviglimus. Appearance of the tassel on tillers, which is the bulk of the main plant here. Mexicana itself takes, well over 4 months to flower, almost 5 months to flower in Missouri. Parviglimus doesn't flower in Missouri, but Northern teosinte does. Note, Gaspé flint doesn't have any tillers. That's why there's no data for the Gaspé flint tillering. The first appearance of silks, this looks just like teosinte. It's amazing. Gaspé flint flowers within, a little bit less than a month and a half. Northern flint, is less than 3 months. So this is pretty good. Parviglimus doesn't make silks in Missouri, and Mexicana hadn't at the time that this data set was put together. And then height in the field, Gaspé flint, if you've worked with that, it's a little runt. It's about 2 feet tall total. The northern teosinte is pretty decent, whereas, of course, the Mexicana is gigantic, and the Parviglumis doesn't make a tassel, so you can't find the flag leaf, so you don't know the height, but it's big. And then tiller numbers, you can see the large number of tillers. Gaspé Flint doesn't make any tillers. It has only the main stock. That's why the number is 1 there. And so we have a nice candidate gene here, or nice candidate material here to work with. So we also collected some leaf tissue of, 18 northern teosinte plants. The couple different accessions of Gaspé that we had in our experiments this summer. Some Parviglumis and Mexicana, and we did the basic Agriplex SNP assay just because we had some empty cells to fill up in those plates that we were talking about earlier. And so some of the questions that we wanted to answer here: how much heterozygosity is in this northern teosinte population? If Tom is going to use this for his de novo domestication, are we going to have any challenges with, are they going to be segregating for things? We need to figure out. Which loci we were fixed during adaptation to the long days flowering times? It's probably VGT1 and ZMTCT. What other loci? We should see signals of, introgressions and things like that. And then Tom is also doing some Sanger sequencing to figure out which allele of TIN1, it's another tillering gene in the northern teosinte, because it can interfere with TB1. And so we need to know what the alleles are present in this population. So here we are, so much corn, so little time. I didn't even talk about the Circa project, which we could probably talk about for a full hour. We didn't talk about genomes to fields or SynAdapt, which is an ongoing project that people probably in this call have heard about. So this is just a sampling of what's going on in the group. The group being a very large group of people. Susan's been my long-term technician for many years. Nancy's been with me for 7 years now as a research scientist and postdoc before that. Sarah, Zach, Melissa, Tom are all grad students that have been in the lab. And Ella, who I didn't talk about today, is a new graduate student this year. She will probably be taking over many of the projects that were started by Sarah, but in the context of the Circa project, again, which I didn't talk about. We've had many different undergrads over the years. These are just a sample, I think most of them, since about 2020. We had at one time 2 Garretts, the Garrettses, and so one of them has a 2T and one is a 1T, and we found a sign that has 2 Ts, and so they were, fighting about that. We have a really good time with my group. There's a lot of goofing around, but a lot of hard work doing all this corn. And so, of course, I have great collaborators for the Heirlooms project, the whiskey project, the asparagine and acrylamide, and then the pigments. And so with that, I will take any questions. I see there are a lot of questions in the chat.
Oh yeah, it's been very lively. Thank you for the fantastic talk, Sherry. Let's see. I think at least going by the chat, SE might have been the first person to raise a question. Going about.
Sorry, Sherry, that was me. This is Jeff. You showed the PC of the phenotypes, and I was wondering what PC2 that separated the Southwest material was.
Yeah, we don't know exactly. I'm assuming once we get some genotyping data, maybe we can correlate some particular genes or something like that in there as well. There even seems to be something like that. The desert Southwest germplasm is always different whenever you see, whenever you do principal components and things like that in large germplasm collections that are very diverse. I wouldn't be surprised that there's some kind of more ancestral, teosinte or primitive land races coming in from the Southwest somehow. I don't really know how that would manifest, but yeah. Okay.
Maybe just to tack on like an additional question to that, because I was actually kind of curious about this too. At least some of like, I have some field sites in like Sedona and like Moab, and the one thing that kind of comes to mind when you're out in that region is that it is a very, very different abiotic environment than the Midwest. And, there's a number of sort of xeric traits that you could automatically see would make a lot of sense. Like, there could be cuticle deposition, root shoot ratio could be really different. Stomata could be doing vastly different behaviors to
Right.
the
Yeah.
best cultivars. And I guess I was wondering, like, have you been thinking about that sort of like ecophysiological component?
One cannot think of everything.
Oh,
Is the quick
yeah.
answer. So we talked within the project within the project, the Heirlooms project, we're doing how many traits could we do and how crazy would it be to do this? And do we really have to do that on 900 entries per year location? And so it's important to remember that when you do these principal components, that's only reflecting the data that you're putting into it. And so I remember the quick answer to Jeff's question. The traits that were correlated with the principal component loadings and all that. I believe it's the tassel architecture that is driving that principal component too. And so it's probably the northern Flint, bigger tassel, like the fewer branches, and maybe the larger tassels and things like that. And so, yes, there probably is a xeric component, but we didn't measure that. And so that's not driving that's principal component two in any way. If you'd like to measure things, you let me know and I'll send you some seed.
I need to convince Andrew to let me grow it first. Let's see. I think I'll go ahead and I'll skip over the next question. Ed, do you want to go over yours with tortilla quality?
Yeah, well, sure. I was just kind of wondering, are you doing pasting content qualities, and is that still a good measure for the gelatinization and things like that? And
Right. So gelatinization is really important in tortillas, and so we are very interested in that. It's not a cheap trait to take, as you remember from the good old
Yeah.
days. We don't have said machine to do that here that I'm aware of. Maybe they do over in food sciences, and I just need to find the right people. I think that pasting properties are going to be very important.
right,
Yep.
okay.
Yep,
And,
absolutely.
and have you guys mapped anything else to sugary one other than sweet corn? I mean, we still have that old hypothesis that there's absolutely something going on in sugary one, that's not is pre-sweet corn.
We haven't done a lot of GWAS on food traits yet because throughput's just not quite there yet. If we ever get our USDA building, which has been constructed and deconstructed and complicated by a lot of things right now, that we're going to have a food lab in there. And so hopefully we'll be able to be much higher throughput, because right now we do the tortilla stuff in our lab space where everybody else has got their lunches and their office cubicles and things. And it turns out to be a huge mess. Okay, let's see. LE. Genome assemblies for the ASN3. Where that's something that Sarah is working on now. Oh yeah, she replied. Yes. So
yeah. Thank you for an awesome presentation also, by the way. I just mentioned it because that's what I had to do with the GA loci, because they're kind of pretty
wonky.
messy in terms of presence absence. But then because the presence absence is very strange, there seems to be very reduced crossover between haplotypes in the region. So the flanking regions ended up being pretty good fingerprinting. But some of the presences were very different from each other, and some of the absences were very different from each other. So you would need like the long read information first, and then
Right.
you can go back and use the
Right.
SNP data. Yeah.
So we've had a couple advisor meetings with Ruthie and going through all the possible combinations. We've even invoked Matt Hufford on some of these conversations about how are these HapMap3 data sets built? I think HapMap3 was still built on blasting back to B73. And so anything that wasn't in B73 gets put in the trash bin. But it can be weighted through and sorted through and looked through and things like that. Cinta, and I don't remember exactly who was doing the genotyping or the sequencing of our two parents for that population. So we have something in the works to look at those specifically, and we'll look at those regions. Sarah looked at LD in the region, and LD does break down very fast. And so it's really quite weird. I don't know that the lack of recombination and having a large LD block and using flanking SNPs and things, I don't know that that is the case here.
Okay, that makes sense. Well, good luck. It sounds complicated.
Hey Sarah, great talk, Ms. Jiniang. So regarding the free asparagine, so for the GWAS, I think Ed and I have a discussion about it in the chat. So we think might be, so because asparagine synthesis is a very important gene in the metabolic process. So it's not surprising if that gene is not segregating in the modern maize population, or possibly be a rare allele. In
Right.
that case, the power might be low. But I'm also
Yes.
thinking, so this asparagine and also glutamate, other amino acids, is nitrogen related, might have something to do with the nitrogen metabolism or nitrogen use efficiency even. Maybe you want to also mapping the asparagine relative to the
Yes.
N-containing, nitrogen-containing secondary metabolites or non-nitrogen containing secondary metabolites. See if that
Absolutely, yes. Yeah. Okay, so let's see, that was a long list of things. So we did look at power and alleles and things like that. So Matt suggested we look at the SNPversity, and there are SNPs. Now we have, this is like just last week, I think. Sarah got some SNPs that were in ASN3, and she did the GWAS on them, and they're not significant. But in the meantime, we were thinking about whether this was a domestication gene and there's maybe no variation because it's fixed and everything are very important. So we went back through and we looked at Matt Hufford's, I think it was the 2012 paper, looking at the domestication loci and things like that for selective sweeps. And it wasn't on that list of those genes. We are very interested in the nitrogen overall, and that's the CIRCA part of the project that I didn't have any time to talk about today. The CIRCA project and our part of the project is to try to reduce the nitrogen in the grain by 50%. And so that's how we started to get some of these ASN3 knockouts and things like that, started to get those created. We expect that some of the knocking them out and having variation that have very large effects is probably not good for the fitness of the plant and or the seed. If you look at the expression levels, I'm again not the transcriptomicist you're looking for, but if you look at the data sets that are out there already, why can't I think of it right now? Okay, if you go to Maize GDB and there's a tool, you can look for the transcriptomics. I can't think of it right now. Anyway, if you look at that, you can see that the 3 asparagine synthetase genes have different expression patterns across the plant. In CIRCA, we tried to make a knockout, a CRISPR targeting, 4 guide RNAs targeting all 4 at the same time. And no surprise there that we didn't get anything out at the end, that only a couple of the genes were edited at the same time. So we're going to try to sort that out once we get those. That's the preliminary reports back from Wisconsin. Looking at the glutamate parts of things, yes, Sarah has a whole nother project looking at all the amino acids and looking at all the families, including the relationships with glutamate and things like that, that I didn't have any time to talk about today and things like that.
Just a quick follow-up. Maybe not simply knockout, maybe you need to reintroduce the teosinte allele.
You know what?
Very hard.
I still hope that teosinte is going to have a place in modern agriculture. It might take a while. Okay, I know we're running out of time here. Let me see if there's more questions here. We already talked about Jeff's question about principal component 2. Yeah, oh, and you also talked about like filters and things like that. Yes, we worried about if there was only like 1 or 2 alleles, and then perhaps it got filtered out. Usually use a 0.05 or 5% for filtering. We checked on that. That wasn't the case. That's when we found out there were no SNPs in asparagine synthetase 3, things like that. So we're looking at, yes. Okay. Sarah has added some text there about the SNPversity, things like that, about how the SNPs were missense mutations. So, we don't really know. Okay. Is there any question that I missed that people really want me to answer?
Looks like we did a pretty good run at most of them. Does anybody have any pressing last minute questions?
Hi. Yeah, Jonathan from the Buckley Lab here, Sherry. I would just keep an eye out also for transporters that might be controlling the flow of asparagine. You probably know, but I'm looking at a senescence data set, and I was looking for asparagine synthetase to be very upregulated throughout senescence to export, nitrogen to the grain, and I didn't find it. So I think something else is controlling the activity of this enzyme. Yeah, it might be either the synthesis of glutamate or glutamine and one of the transporters. And I would look for genes that are phloem specific because that tissue is controlling where this nitrogen goes. That's like what I keep thinking lately. Just a suggestion.
Yep. Sarah, oops, there's an echo. Sarah went to the Canvas meeting last week and talked to Sakiko, and obviously she's really interested in the nitrogen transporters, and so we really need to make a better connection with her on some of these things. And the worldview that we're kind of looking at the asparagine specifically was because it's a I shouldn't say dual use. It has two purposes for the asparagine in the acrylamide story, and then also as a major nitrogen transporter. We know that that's not the whole story, and by taking care of asparagine in the grain, we're not going to reduce nitrogen by 50%. Sorry, Ed, that's not our only target. Don't you worry. That was just something that Sarah was already a grad student working on this project, and we thought, well, we can kind of roll a lot of these things in together. And make it multi-purpose, I should say. So yes, we're aware that it's not just about asparaginase, the tastes and things like that.
Thanks.
I should have, yeah, go ahead.
Very inspiring talk. Thank you.
This is what you can talk about when you get farther in your career, and you can have all these big ideas, and you can point to what all the people did and things like that. I have a really great group of people that I'm trying to not let them graduate and move on. So if you're looking for a postdoc, Sarah Fitzsimmons is going to be graduating in May, but I totally recommend you don't take her away from me because I'll be very mad at you. Just kidding. If you want to hire Sarah, you should talk with her. I know, you got to let your little baby birds fly away. I know.
All right. Well, I guess are there any other questions or ideas?
Did you know how many slides I went through in that short time? I amazed myself. This was crazy. That was 65 slides.
Very democratic.
All right.
Well, thank you so much, Sherry. It was great having you here. Really glad that you could
Back.
this semester.
Yeah.
And yeah, this should be online in a couple days if people missed it. So yeah, thanks everybody for coming. Thank you for showing up, Sherry.
Yep.
And we'll see everybody next week.
Okay. Bye-bye, everyone.